Welcome to my projectbazaar.com. Yeah, let us discuss about the prediction of leaf disease using segmentation with hierarchical clustering. Yeah, let us discuss about the abstract of the product. So the large number of exciting plant species in the world makes human identification of them tedious and time consuming, particularly for non-expected stakeholders such as land managers, foresters, and then gardeners, etc. So hence an automatic plant identification tool should be speed up the plant species identification task. So this identification tool may be useful even our experienced botanist. Plant identification is based on the observation of its organ, like that is buds, leaves, fruits, stems, etc. So in this proposed work, so we focus on the shape of the leaf and shape based on the approaches of leaf recognition. To describe the shape of a leaf, one can develop the specific approach of adapt generic space retrieval method to the particular case of leaves. So in this work, we propose with the idea of imaging technology, the plant diseases detection systems automatically detect the symptoms that appear on leaves and stem of a plant and help is cultivating healthy leaf in a farm. So these systems monitor the plant such as the leaves, stem, any variation observed from its characteristics of features. So variation will be automatically identified and also it will be informed to the user. So the leaf disease, it will be must to predict the values for the particular farmer or like that. So this paper and then this work provides an evaluate study on existing disease detection systems in a leaf. So whatever it may be, leaf disease is must to predict in the agriculture or like garden like that. So prevent the disease from that. So these are the overall process of the abstract. Yeah, let us discuss about the flow diagram of the process. So this is the overall flow diagram. Here we are using the input image. So that is the input image what with the leaf detection and then pre-processing step, segmentation, feature extraction, classification and organized output. So these are the 6 models in the basics of data flow diagram of the process. So first of all, the input image. So in this input image we taking the disease leaf. So the basic MATLAB data structure is an array, an order to set of real or complex complex elements. An array is naturally suited to the representation of images, real valued ordered sets of colors or intensity data. So in the MATLAB workspace, most of images are represented as two-dimensional arrays in which each the element of the matrix correspond in a single image it should be displayed. And then in the pre-processing step, so here we are using the pre-processing step, filter and also the RGB to HSV color transformation. So in this proposed work, the RGB images were converted into HSV color space representation. In the RGB model, each color appears in its primal spectral components of red, green, blue, and this model is based on the Cartesian coordinate system. So the RGB model matches to the human eye in such as describing that. To avoid these limitations, the accurate RGB images are converted into the HSV format. So and then segmentation process. So in the segmentation process, applying 3 types of clustering. One is a k-means cluster, again one unaffected cluster, and then also a masking process, that is a thresholding base. So in the k-means clustering, we reduce the 3 type of k-means clustering is used, and then if affect cluster, this is a masking process. The disease it should be identified and it should be masked. And then threshold base, so like the threshold base means that's zeros or ones. That's a representation of threshold base algorithm of the process. So by using the thresholding process, here we using the in this work, the leaf mass and damage area mass that's generated are taken further out the analysis. So the damage area mass was multiplied with the resized RGB images obtained after the pre-processing step. So since the mass contains only ones and zeros, and in the mass resist portion of the leaves and having the values of ones, when this image is displayed and then it should be multiplied with the RGB image, only this images portions will come out as a result of multiplication. And then in the feature extraction process, to extract the feature by using the GLCM. So that is a gray level co-occurrence matrix. So the gray level co-occurrence matrix image is given image. In the first step, the GLCM by calculating how often a pixel with gray level value is occurs horizontally adjacent to a pixel with the value of G. So each element will be calculated I, J in the GLCM specifics number of time that the pixels with value occurred. So horizontally it specifies the values of 10. So the texture features can be extracted using the GLCM values. And then classification. So in the classification process, here we are using three classification. One is a neural network, another one is a SVM, and another one is a KNN process. So three classification is used to identify the process, to identify what type of disease will be fine, and then finally recognize output. It should be the performance measure like accuracy, specificity. So for the classification process, so these are the overall flow diagram of the process. Yeah, now we'll let us discuss about the running procedure for the product. So click the new file and then run. So here we're using the disease. What type of disease? So I will select a data set. The many images will display. So I will click one type of disease identified leaf. So I will click that touch and then click open. So the process, it should be displayed. Whatever it may be, all the values it should be displayed. Yeah, I will select this on input. So this is the one type of input of the leaf. So and then the next figure is identified. So in the next figure, so this is the original color, red channel, green channel. So image with salt and pepper noise, noisy, red, green, blue, and then restored an image for noise free, red, green, blue. So these are the different types of images. And then the next figure, so this is the RGB and HSI. So here we are converting RGC. Already I told in the flow diagram. So RGB is converted into the HSI of the process. And then the next figure, so this is the RGB and HSI of the process. So I will click that. Here I will using yeah, so this is the RGB image, and then HSI equalize. So hue, HSI means hue, saturation, intensity. So these are the three types of hue and then saturation, intensity of the process. So to identify the leaf disease of the process. And then in the next figure, yeah, this is the so in this figure, so original color image, gray, binarized, noise free RGB. So there is a green, blue, and then green pixel mask and mask the RGB. So I already told them in the segmentation process there are two types. One is a thresholding and another one is a masking process. So in the masking process, the disease and leaf, it should be masked by using the RGB of the process. And then the next figure, so in this next figure, that the mask by using the mask image, again the same process, hue image, saturation, and then saturation clustering of the process. So hue, saturation, intensity of the process. Again regression plot. So the regression plot by using the neural network of the process. So this is the target and this is the output. So both are the target and regression to the value the converter. And then error surface. So in the error surface region, the leaf only identify the disease will be calculated. So the error surface is sum square error in x-axis, weighted in y-axis, z-axis in bias. So this is the error phase surface. Finally calculating disease identified as the anther constant infection. So this is the warning dialog box will be applied. So this is for one type of classification, and then another two type of classification. All the process it will be displayed. So in the another type of classification, so that it will be this accuracy, so sensitivity of KNN, and then neural network classifier. So this is a multi-SVM and then KNN process. Similarly for comparing the graph, so that is the existing and proposed of the value. So accuracy, error rate, sensitivity, specificity, and the same process by calculating by using the SVM process, so existing and proposed. So all the methods by calculating the three methods. So these are the overall running procedure for the process. Thank you for listening this video.